Welcome to the deep dive. Today we're undertaking a really critical mission. We're going to distill the standards of care in laboratory diagnosis.
That's right.
We've been digging into some key clinical chemical sources to give you a genuine shortcut to understanding where this high-stakes system of medical testing most often fails.
And this is just absolutely foundational material for anyone in healthcare. Our sources all guide us with that primary ethical principle, above all, do no harm.
Right.
And what's so crucial to understand is that minimizing diagnostic error, it means looking at the entire process. It's not just about the machines.
That's where the real complexity is, isn't it? We all have this picture of sleek, automated instruments, but the sources make it clear that the automation is, well, it's irrelevant if the initial sample is compromised. So what's the true focus of error prevention here?
It's the preanalytical and the postanalytical phases. Most of these failures actually happen outside the laboratory walls.
So when the blood is being collected or when the results are reported?
Exactly. When it's being collected, transcoded, or interpreted. True quality management requires aggressive interdisciplinary collaboration. We're talking between nursing, phlebotomy, informatics, and the lab staff.
So we're going to cover some of those common high-frequency mistakes.
The ones made daily across thousands of institutions, yes.
Okay, let's start with segment 1, the critical foundation. This is all about specimen integrity and pre-analytical failure.
The very beginning of the process.
And the first layer of defense is purely human. It's just verifying identity.
And the standard of care here is, well, it's non-negotiable. It's designed to completely eliminate mix-ups. You must use a minimum of 2 unique patient identifiers.
So full name, date of birth, something like that?
Usually full name, date of birth, or the medical record number, the MRN. And here's the kicker. Labeling of the collection tubes has to happen immediately after collection in the patient's presence.
Okay, so why is that last rule so strict? I mean, why can't a phlebotomist just label the tubes once they get back to the workstation?
Because pre-labeling is a prime opportunity for catastrophic error. It just is.
A mix-up waiting to happen.
Absolutely. Imagine a phlebotomist prepares a whole rack of tubes for 3 different patients. They get distracted, they come back, and they use the wrong tube on a patient.
And that result will now follow that patient forever.
Indefinitely. The requirement to label immediately while you're confirming the identifiers with the patient is really the only robust safeguard we have against misidentification.
The sources have some great anecdotes that illustrate how policies that seem like bureaucratic hurdles are actually critical safety nets.
Yeah.
That delayed blood gas case is a perfect example.
It is. So a nurse collected a blood gas sample, but failed to label it right there. So the laboratory correctly rejected the specimen based on policy.
Okay.
So a second collection was attempted. But that sample, it arrived at the lab delayed. We're talking more than 45 minutes after collection, and it was at room temperature, not on ice.
And what happened to the results then?
Well, for highly volatile analytes like blood gases, pH, ionized calcium, the sample just degrades incredibly rapidly with time and temperature.
So the data is garbage.
It's completely unreliable. The lab had to cancel those results, which then forced the healthcare team to collect a 4th sample.
A 4th one.
A 4th. And while the ordering physician was, irate about the obstructive policy, the lab's strict adherence prevented the release of misleading data that could have driven inappropriate, even fatal medical intervention.
So those policies, they really are there to protect the patient from bad data.
That's their only purpose.
Okay, moving beyond identification, collection errors can ruin the chemical integrity of the specimen itself. Let's talk about additive contamination.
The simplest case is just using the wrong tube. Imagine a test requires a red top tube. That means no additive. But instead, the lab gets a purple top tube.
Which has EDTA in it.
Right, which contains EDTA. And EDTA is a powerful anticoagulant that works by chelating or binding calcium ions.
So any test that needs calcium is going to be thrown off.
Any assay that relies on free calcium, coagulation studies, certain trace metal tests, even a free T4 immunoassay. The EDTA interference just renders the result chemically invalid. It's an immediate recollection. No other option.
That's pure chemistry preventing the analysis. Now, what about that pre-analytical failure of using an indwelling IV catheter for collection? That's known to cause dilution, contamination.
Yeah, you hit on a really high-risk area. Since collecting blood through an IV line can lead to unreliable results, the process has to be strictly controlled.
So what's the standard of care to mitigate that risk?
Okay, so if a clinician determines that collection through an indwelling line is absolutely necessary, maybe because of truly poor vascular access or a high bleeding risk, 3 specific requirements must be met.
Okay, what are they?
First, it must be based on a physician order, one that specifically acknowledges the risks. Second, it can only be performed on patients where poor access justifies that risk.
And that's the third.
And third, the result must include a specimen comment, something that notes it was collected through line.
Why is that so important?
Because it alerts the ordering provider to the potential for dilution or contamination. It helps them troubleshoot the result. It gives context.
So those 3 steps ensure that if contamination does happen, the result isn't just treated as gospel.
Precise.
Speaking of compromised samples, what about temperature abuse? Using outdoor specimen drop boxes seems like a minor logistics issue, but it causes huge problems.
It's a massive problem, especially for outreach programs. If a sample freezes, it causes hemolysis.
The red blood cells burst.
They burst, and that releases massive amounts of intracellular components into the serum. So it artificially elevates potassium, LDH lactate, dehydrogenase, and certain enzymes.
And what about heat?
If the sample overheats, accelerated cellular metabolism changes glucose and pH levels, the lab has to provide clear, environmentally controlled recommendations for transport. It's the only way to ensure analyte stability.
Okay, let's transition into segment 2. Inside the lab, we're focusing on analytical pitfalls, shortcuts, and quality control. Now, the quality control process is the lab's internal self-check, and the source describes a common and pretty dangerous shortcut that technologists sometimes take.
This is the QC failure trap. A core requirement is running QC samples at 2 different concentrations to verify that the instrument is accurate across its whole measurement range.
Okay, so what's the shortcut?
The shortcut happens when a control fails, and the technologist just repeatedly runs that same sample, maybe 5 times, until it randomly, by chance, falls just inside the acceptable range.
And then they release patient results.
It's essentially trying to hide a system flaw.
So what should they be doing instead?
They need to pause. They need to stop. If QC samples at 2 concentrations fail, especially if they both fail on the same side of the mean, that signals a systemic failure.
It's not random.
It is not random chance. It demands immediate instrument troubleshooting. Checking for reagent degradation, recalibrating, maybe checking the light source. Repetition is never a substitute for fixing the root cause.
Now, point-of-care testing POCT is often marketed as a solution to increase efficiency. But the sources show it can actually create more management complexity.
It does, because POCT implementation often tries to solve complex scheduling or staffing failures, rather than being driven by pure medical necessity.
I think I know the case you're referring to.
The cardiovascular unit case. They implemented POCT for electrolytes, and it increased delays.
Why? How did that happen?
Because the staff just added the POCT testing burden. All the complex QC, the documentation, proficiency testing onto an already uncoordinated system. The standard is clear. POCT must be driven by an immediate patient medical need, not as a shortcut to bypass established system management failures.
And there are serious regulatory requirements for the operators of these decentralized devices, too. I mean, regarding competency.
Yes. Competency assessment has to be rigorous and specific to the device. You can't just assume a general laboratory certification covers it.
The sources highlighted a really shocking scenario with this.
The legally blind operator attempting to run a POCT device, yes. This operator, because they had difficulty reading the display in the keypad, was making serious identification errors. A supervisor has to verify the operator's ability to safely and correctly operate the specific device they're assigned to.
And if they can't,
their privileges must be suspended immediately. No question.
Before we move on, there's a critical safety issue tied to the analytical process we need to cover. First, the mechanical hazard, centrifugation.
Centrifugation is, it's deceptively hazardous. We saw the case where an incorrect RPM calculation caused a high-speed centrifuge to run too fast.
Generating a lot of friction.
A ton of friction. This caused the temperature inside the rotor chamber to rise dramatically up to 45, 50 degrees Celsius, and that compromised all the heat-sensitive analytes in the samples.
So proper calculation is essential.
Calculation of G-force and careful monitoring of temperature. It's essential to prevent that kind of degradation and sample loss.
And then there's the severe laboratory safety hazard involving improperly handled biological waste. The explosive specimen.
Oh, yeah.
You have to tell the listener about the fecal fat specimen.
This case is famous, or maybe infamous, within lab safety circles. A specimen for fecal fat analysis was collected in an unvented paint can.
An unvented paint can.
Exactly. And because it was left in a hot outdoor drop box over a long holiday weekend, bacterial gas production just went into overdrive, generating immense pressure inside the can.
I can see where this is going.
When the can was brought into the processing area, it violently exploded.
You're kidding.
No. The biological material was aerosolized. It was embedded into every single surface of the processing area. They had to close the room down and sandblast it.
Wait, sandblasted?
Sandblasted. To ensure complete biohazard remediation, the standard of care is absolutely clear. High gas-producing specimens, especially those transported over extended periods, must use vented containers. Period.
Wow. That illustrates the stakes perfectly. Okay, let's move into segment 3, informatics. We're covering the LIS, the EMR, and the postanalytical breakdown.
Right.
So for our listeners who might not be familiar with that jargon, what are we talking about here?
So the LIS is the Laboratory Information System. That's the specialized software where testing is ordered and results are processed.
And the EMR?
The EMR is the electronic medical record. It's the patient's permanent chart where clinicians view those results. And failures at this interface are incredibly common.
And one of the simplest failures starts right at the beginning. Test naming confusion.
Which leads to overordering. This happens all the time when new test methodologies are introduced. For example, if you introduce a high-sensitivity CRP.
Which is for cardiac risk.
Correct.
Yeah.
The naming in the LIS has to carefully distinguish it from the standard CRP used for inflammation or sepsis. If the names are confusing, clinicians will misapply the test.
And that overordering leads directly into what the sources call the incidental finding cascade. Tell us about the resident who ordered the entire thyroid panel.
Right, so this resident, unsure of the correct way to evaluate hypothyroidism, ordered the whole suite. TSH, FT4, the rarely used T3RU, just everything.
When the standard is sequential.
The standard is the sequential strategy. You start with TSH, and then you reflex to FT4 only if the TSH is abnormal. It's clinically sound and cost-effective. By ordering the whole panel, they found a detectable level of thyroid-binding inhibitory immunoglobulin.
So finding this immunoglobulin, even if it's not clinically significant, it forces further action, right?
Precisely. Even though the titer wasn't high enough to meet diagnostic criteria, just the fact that an abnormal result appeared, it caused an additional unnecessary workup for autoimmune disease.
So overutilization increases costs and can be misleading.
It absolutely burdens the patient and the system.
The final post-analytical failures happen when results are viewed. And the case of the suppressed specimen comment in the EMR is just terrifying.
This is a huge design flaw in how EMRs can present data. A critical care resident was viewing a low potassium result, 2.9 millimole, in a tabular summary view.
Just a table of numbers.
Exactly.
Yeah.
And crucially, the specimen comment, which stated, specimen potentially contaminated, drawn above PIV line, was hidden. It was suppressed, or it required a separate click to even view it.
Meaning the resident was treating that low potassium as a genuine finding that required intervention.
Exactly. The standard of care demands that EMR systems actively draw the user's attention to any critical comment associated with a lab result, especially if the comment relates to specimen integrity. Hiding those vital details risks catastrophic misinterpretation.
And sometimes the interface failure isn't suppression, it's outright data corruption.
Interface failures require ongoing monitoring, not just a one-time verification. We've seen data corruption where a CA125 result over 100,000 was just displayed as too big because the legacy interface couldn't handle 6 digits.
That's bad.
Even worse are decimal errors. A truly high-stakes potassium value of 4.0 millimol was displayed as 0.4. Imagine the clinical action taken based on that decimal shift. It's an immediate threat to life.
Okay, let's move to segment 4, specialized context. Starting with therapeutic drug monitoring and toxicology. These areas seem to involve the highest level of patient deception.
They really do. So TDM involves monitoring drug levels, things like vancomycin or lithium, while toxicology screens for drugs of abuse. And patients use 3 main methods to generate false negatives.
Which are?
Dilution, so drinking excessive water or using diuretics. Substitution, using synthetic or borrowed urine. And adulteration.
How sophisticated is this adulteration?
Very. They use everything from dish soap to acids. Adulterants work by interfering with the immunoassay. For example, some common household acids will cause a structural change in the antibodies used in the assay.
Leading to a false negative.
Right. But conversely, some adulterants, like certain oxidants, can actually cause a false positive by mimicking the analyte. That's why the lab has to check integrity indicators like creatinine, osmolality, and pH.
And the physician has to be aware that even legal over-the-counter drugs can create false positives.
Absolutely. The clinical standard is that you should rarely, if ever, treat a patient solely based on a screening immunoassay result. Common cold medicines with ephedrine can cross-react with amphetamine assays.
Okay. Any other examples?
Yeah, dextromethorphan, which is in a lot of cough syrups, can flag a false positive for PCP assays. Confirmatory testing, usually with a highly specific method like GCMS, is essential to rule these out.
Finally, let's revisit the critical value responsibility. This is governed by specific regulations like CLIA. What's the non-negotiable legal imperative here?
Critical values. Those results that represent an immediate life-threatening situation, they demand immediate interruptive contact. The lab must reach a live person.
Usually the ordering physician.
Or their designee. And crucially, that person must then verbally read back the result to the technologist to confirm accurate transmission and comprehension. It's a legal chain of custody for information.
We saw the friction that can happen when physicians try to skirt this, like in the vancomycin case.
That case, yeah. It involved the vancomycin test ordered by a physician as a courtesy for a patient who was already discharged. When the lab called with a critical high value, the physician refused the call.
Claiming it wasn't his responsibility.
Right. It's a clear breach of professional expectation. Ordering physicians must, legally and professionally, take responsibility for follow-up on tests ordered under their name.
And what about outreach clients asking to leave messages?
A lab cannot legally comply with a request to leave critical results on an answer machine. They have to maintain that immediate, live communication link. It's required.
Okay, so to synthesize everything we've discussed.
I'd say the quality management in clinical chemistry, it extends so far beyond just the analytical instrument. It requires continuous vigilance across the entire system.
From start to finish.
From the phlebotomist ensuring 2 unique identifiers are used, to the technologist troubleshooting a systemic QC failure, all the way to the EMR that's actively displaying necessary specimen comments. True effectiveness depends on collaboration, communication, and constantly questioning assumptions.
That provides a really comprehensive picture. So, to reinforce the standards of care for you, our learner, let's end with a short review exercise based on one of the pitfalls we covered earlier.
Excellent idea.
We discussed the high risk of contamination when collecting blood through an indwelling ABEA catheter. So, based on our discussion, what 3 specific requirements must be met if a clinician determines that collecting a specimen through an indwelling catheter is absolutely necessary?
Think about the steps required to validate that risk. The 3 requirements we talked about are, first, the collection has to be based on a specific physician order that acknowledges the risk. Second, it can only be done on patients with truly compromised venous access or high bleeding risk.
Right.
And third, the laboratory result has to include a comment, something like collected through line, to alert the ordering provider to the potential for dilution or contamination. That helps them with the clinical action.
If those 3 steps aren't followed, the quality chain is broken right there.
It is.
And finally, building on those concepts of cost, utilization, and test menus, consider this provocative thought. When hospital outreach programs prioritize aggressive business growth by facilitating any test a physician wants, how does that financial incentive often undermine the clinical goal of limiting unnecessary test utilization and controlling costs for the rest of the health system?
Something to mull over as you integrate these standards of care into your practice.